The pentose phosphate pathway is a metabolic pathway that's found in many cells in the human body. It's particularly enriched in liver cells, red blood cells, adipose tissue cells, the adrenal cortex cells, also liver cells. These types of cells are very much enriched in pentose phosphate pathway enzymes. Pathway also exists by other names. One of them is called the hexose monophosphate pathway, or the hexomonophosphate pathway, HMP. It's also known as the phosphogluconate pathway because one of the intermediates in the pathway is phosphogluconate. And the net reaction of the pentose phosphate pathway, a very critical reaction, works hand in hand with glycolysis. For example, if glycolysis doesn't work, the pentose phosphate pathway will start to really kick in. But there are some differences between the pentose phosphate pathway and glycolysis. For one, the products that we produce, now shown here is the net reaction of the pentose phosphate pathway. We take 3 glucose 6-phosphate molecules. That glucose 6-phosphate can come from glucose and the hexokinase reaction. 3 glucose 6-phosphates, we get 6 NADPHs. Now, in Biochemistry 1, we talked about the role of NADHs. Now we're talking about a different cofactor. The only difference is that inorganic phosphate. But from 3 glucose 6-phosphates, we get 6 of the nicotine adenine dinucleotide, the phosphorylated form. And we get 2 molecules of fructose 6-phosphate and 1 molecule of glyceraldehyde 3-phosphate can go and get shunted into glycolysis. In fact, many people call this the hexose monophosphate shunt because the fructose 6-phosphate and the glyceraldehyde 3-phosphate can immediately get recycled back into the glycolytic pathway or even to the gluconeogenic pathway. Generation of this reducing equivalent known as NADPH. This is very important in the cell because it's primarily involved in what is known as reductive biosynthesis. There's a number of anabolic pathways, that is, pathways that build carbons and build macromolecules that require reductions in each of the enzymatic steps in the pathway. Well, with those reductions comes oxidations of cofactors, and it's this NADPH that's going to serve as the reducing agent itself getting oxidized to allow biosynthesis of important molecules to occur. Here's nicotinamide. The dinucleotide comes here with this nitrogenous base as well as this nitrogenous base. And really the chemistry happens here in terms of oxidation and reduction. More specifically, the hydride transfer occurs at the number 4 carbon in the reduced form. However, if you look here at this sugar side of the NADH or NADPH molecule, you have a portion here that could either be an H, in which case it's going to be NADH, and the oxidized form is NAD+, or that X could be an inorganic phosphate. In that case, the phosphate would go here, and this would be NADPH. The oxidized form would be NADP+. The important thing to realize here is that NADH and NADPH, these two reduced cofactors or reducing equivalents, are not the same. You can look at the ratios of these in a typical cell, and you'll find that the NAD plus over the NADH ratio is about 1,000 to 1. So you have the oxidized form that is ready and willing. It's particularly important for those dehydrogenation reactions. Very familiar with dehydrogenases from glycolysis. So the cell will have an ample supply of the oxidized form of NAD plus versus the NADH. Not so when you compare it to the phosphorylated form of the cofactor. The NAD plus oxidized form, its ratio to the fully reduced NADPH is about 0.1. So actually it's a 1 to 10 ratio. For every 1 molecule of NAD plus, you'll get 10 molecules that is in NADPH form. So different ratios and really different functionalities. These are not interchangeable. The cytoplasmic pool has different concentrations of each of these cofactors. Going back to NADPH, some of the things that differentiate it between this and NADH is this concept of reductive biosynthesis. So the first thing that we want to know or want to realize is that NADPH is important for reductive biosynthesis. Some of these things that get biosynthesized are fatty acids, cholesterol, so steroids are important in this. Neurotransmitters get biosynthesized through a reductive biosynthetic pathway, so that means they will require NADPH as a cofactor for its synthesis. And also RNA and DNA, nucleotide biosynthesis. And so the pentose phosphate pathway is instrumental in making this, which in turn is important for making these guys. Another important thing for NADPH is free radical detoxification. So a lot of times oxygen radicals or peroxide radicals can just form by normal metabolism, normal oxidation. And NADPH, by its abundance, can actually clear some of these egregious oxygen or hydroxide or peroxide radicals. One way it does it is through the cytochrome system, and another way it does it, and which is found plentiful in the cell, is working in concert with this molecule known as glutathione. Glutathione is actually a tetrapeptide, whose structure is shown here. It's a tetrapeptide consisting of 3 amino acids, glycine, cysteine right here in the middle, and then another glycine. So you can see here that cysteine can readily form a disulfide bond. And so you can have glutathione in its reduced form, or you can have glutathione in its disulfide oxidized form. So the oxidized and reduced form gets switched based upon the oxidation and reduction of NADPH. If one gets oxidized, the other is going to get reduced. And as a result of that cycle, free radicals get detoxified. Here's our pentose phosphate pathway that we're going to talk about later on. But here is the NADPH. Okay, so the pentose phosphate pathway is producing NADPH at this step here, and it's also producing it at the third step in the metabolic pathway. Now, the NADPH can work in concert with glutathione. So here's glutathione, the tripeptide, glycine, cysteine, and glycine, existing as a tripeptide, forms a disulfide bond with the 2 cysteines, and that oxidized form of glutathione can get reduced, and when that oxidized form of glutathione gets reduced, it will oxidize these radicals. So in this specific example, it's oxidizing H2O2 to H2O, and that is a good thing because H2O can be cleared into the cell, and the hydrogen peroxide, which can be pretty reactive and is a source for peroxide radicals, is essentially removed or quenched from the cell. So that is one example of the use of glutathione. Now the question is, how do we regenerate the oxidized form of glutathione so that this cycle can continue onward and onward? Well, that's where the NADPH comes in. So you would see here the NADPH gets oxidized to NADP+, which regenerates the oxidized form. The oxidized form can then go ahead and detoxify some of these peroxide or oxygen radicals, and then you essentially have a cycle that's going on here. So it can do this for other types of radicals, not just hydrogen peroxide and peroxide radicals. NADPH at this step and NADPH at this step. So a plentiful store of NADPHs for not only reductive biosynthesis, but also detoxification. Just want to make clear we have 3 glucose 6-phosphate molecules that are going to go in, and that's going to produce 6 NADPHs. That's how the stoichiometry is going to work. I divide the pentose phosphate pathway into 3 stages. The first stage is the redox step. That's where oxidation and reduction occurs. So obviously that first stage is where you're going to make your NADPH, the reducing equivalent. The second stage of the pentose phosphate pathway is what I dub the isomerization and epimerization phase. So you either isomerize or epimerize. Remember epimers? You isomerize or epimerize the resultant molecules that are made from the oxidation, reduction, redox phase. And then the third step of the pentose phosphate pathway, the third stage, is the carbon-carbon transfer phase. So 3 steps: the redox step, oxidation reduction, the isomerization or the epimerization phase, and then the third phase is the carbon-carbon transfer phase. Ribose 5-phosphate. So ribose 5-phosphate is a precursor to making DNA and RNA. In fact, nucleic acids do require a sugar besides a nitrogenous base and an inorganic phosphate. So ribose 5-phosphate fulfills 2 of those 3 requirements. NADPH, which is generated by the pentose phosphate pathway, or the hexose monophosphate shunt, is used to make cholesterol, fatty acids, neurotransmitters, reductive biosynthesis. And this pathway is found in a lot of cells. Many cells are dependent and are enriched in this pentose phosphate pathway. Many consider it as an alternative to glycolysis. You get here fructose 6-phosphate and glyceraldehyde 3-phosphate is one of the products. So if the cell decides to go into a more glycolytic route, it can do that through this pathway. If the cell decides to go to more of a DNA RNA synthetic route, well, it can also do that by the production and generation of ribose 5-phosphate. Glucose 6-phosphate goes to 6-phosphoglucano delta lactone. So let's just take a look at the chemistry here. Here you have the anomeric carbon. The anomeric carbon is getting oxidized to a lactone. Lactone is nothing more than a cyclic ester. So here's our ester. The ester has formed a ring. So and when the ester closes in on itself, like as is shown here, that is called a lactone. So this is a 6-phosphoglucanolactone. The enzyme that catalyzes this reaction is the rate committing or rate limiting step of this pathway. So it commits it to the pentose phosphate pathway, and that is glucose 6-phosphate dehydrogenase. Obviously, if it's a rate committing enzyme or the bottleneck enzyme, it's subject to regulation. And the regulatory effector here is NADP+. The NADP+, the oxidized form of the cofactor, is an allosteric activator of this enzyme. So if the NADP+ concentrations are significantly high, that means one thing, there's a lot of free radical detoxification going on. And it also means another thing. It means the cell needs to increase flux to generate NADPH through this pathway. So NADP+ will be an allosteric activator of glucose 6-phosphate dehydrogenase. The 6-phosphatidylglucanolactone actually can break apart rather easily through a ring opening. If it doesn't do it spontaneously, it does it through the second enzyme in the pathway, and that's 6-phosphoglucanolactonase. That's just a ring opening. That 6-phosphoglucanate is the substrate for the second redox reaction in the pentose phosphate pathway, and that is the generation of ribulose 5-phosphate. So notice here, we actually have the generation of NADPH at this step, the first step, the rate committing step, and then the third step. By the way, this is a decarboxylation reaction as well, the dehydrogenase. So we generate reducing equivalents in the third step and the first step. To get the stoichiometry to actually work, it's 3 molecules of glucose 6-phosphate actually are going to go. So if 3 molecules will go to make 3 molecules of ribulose 5-phosphate, that implies that you will make 3 molecules of NADPH here and 3 molecules of NADPH there. So that means 6 NADPHs. It also means 3 CO2s. So that's included in your net reaction here. So the end part of stage 1, which I consider the redox phase, is the generation of ribulose 5-phosphate, shown here in its structure. Stage 2 is basically the isomerization phase. So the 3 molecules of ribulose 5-phosphate formed from the 3 molecules of glucose 6-phosphate is going to go through a 1-step epimerization. Actually, so we'll go through a 1-step isomerization and a 2-step epimerization. So by that I mean for every 3 molecules that are generated, 1 molecule of this ribulose 5-phosphate will be isomerized to form ribose 5-phosphate. And you can actually see the chemistry here. The chemistry occurs when this ketone gets converted to the aldehyde. That's actually a tautomerization reaction, keto-aldo-tautomerization. So the isomerase takes ribulose 5-phosphate, converts it to ribose 5-phosphate. The other two molecules, remember we make 3 of these, the other 2 molecules are going to be epimerized. And the epimer occurs at the C3 carbon. So the C3 carbon gets switched in its stereochemistry. Good. The C3 epimer of ribulose 5-phosphate, which is xylulose 5-phosphate. So one molecule of this gets isomerized, 2 molecules get converted to its C3 epimer, and as a result of the isomerization and the epimerization phase, we get 1 molecule of ribose 5-phosphate and 2 molecules of xylulose 5-phosphate. So stage 2, highlighted in red, is complete. Now, before we move on to stage 3, the carbon-carbon rearrangement phase, this actually is a stopgap here, right here at this ribose 5-phosphate step. At this ribose 5-phosphate step, the cell can decide to shunt this and shunt it to make DNA and RNA. So again, you have a very important precursor to make nucleic acid right here via the pentose phosphate pathway, particularly if the cell is going to decide it wants to grow or for whatever reason wants to make DNA or RNA, nucleic acids. Let's say the DNA and RNA or the nucleic acids are completely content or no longer really in need. So then we can move on to stage 3. And stage 3 in this pathway is the carbon-carbon rearrangement pathway. We have 3 enzymes here. We have a transketolase, we have a transaldolase, and then we have a transketolase. I want to bring your attention here to the arrows. The arrows in stage 3 are all reversible. Notice the transketolase is reversible, the transaldolase is reversible, and the second transketolase is reversible. So you can go back and forth. You can go back from the gluconeogenic or glycolytic intermediates. You can go back to make the DNA, RNA intermediates, or you can go from the DNA, RNA intermediates in this directionality to make the gluconeogenic or glycolytic intermediates. So that's important. Stage 3, that equilibrium and that reversibility gives the cell some flexibility, whether it wants to go to making DNA, RNA, or whether it wants to just fulfill flux through glycolysis. So let's talk a little bit about these carbon-carbon rearrangement reactions that are. One of these ribose 5-phosphates is going to react with one of these xylulose 5-phosphates. So when that happens, that reaction is catalyzed by a transketolase. So one of these RFPs, R5Ps, ribose 5-phosphate, is going to react with one of these xylulose 5-phosphates, leaving one more away. So the 1-to-1 is going to be catalyzed by a transketolase reaction. And essentially what's happening is that this C double bond O CH2OH, that those 2 carbons are being transferred to the number 1 carbon, or the anomeric carbon, of ribose 5-phosphate to give you a 7-carbon sugar known as pseudoheptulose 7-phosphate. Now when these 2 carbons, the carbon 1 and carbon 2 of xylulose 5-phosphate, go and get transferred to here, you're left with this 3-carbon molecule that's left behind, and that is glyceraldehyde 3-phosphate. So when one molecule of R5P reacts with one molecule of xylulose 5-phosphate, the transketolase reaction gives you pseudoheptulose 7-phosphate, a 7-carbon sugar, and a 3-carbon sugar in the form of glyceraldehyde 3-phosphate. The transketolase, shown here in reaction 6, is a TPP-dependent enzyme. It's an enzyme that requires the cofactor thiamine pyrophosphate, which is one of the reasons why thiamine is very important in our diets. Now the cell wants to move on. In stage 3, we go through a second carbon-carbon rearrangement reaction. In the second carbon-carbon rearrangement reaction, we take these 3 carbons, the C double bond O, and the CH2OH here, and we transfer that to this anomeric carbon of glyceraldehyde 3-phosphate. When we do that, we get fructose 6-phosphate, and the carbons that are left behind is this 4-carbon sugar, erythrose 4-phosphate. This is catalyzed by a transaldolase. The transaldolase actually functions via a Schiff-base intermediate. So there's going to be a amine, it's the epsilon amino group of lysine, that's going to form a Schiff base with this carbonyl group of pseudoheptylose 7-phosphate. When that Schiff base breaks apart, you're going to get the carbon-carbon transfer. A different mechanism from the TPP-dependent transketolase versus the Schiff base-dependent transaldolase. Finally, the last stage of the carbon-carbon rearrangement reaction is taking that xylulose 5-phosphate, that one that was left over from before, and reacting it with erythrose 4-phosphate. That is a transketolase catalyzed reaction dependent upon thiamine pyrophosphate. In this particular case, that C double bond O, CH2OH, is going to get transferred to this anomeric carbon of erythrose 4-phosphate, and you're going to get two molecules. One of them is fructose 6-phosphate, and this leftover xylulose 5-phosphate, by virtue of transferring of this C double bond O CH2OH to erythrose 4-phosphate, gets generated to glyceraldehyde 3-phosphate. So one of these ribose 5-phosphates reacts with one of these xylulose 5-phosphates, and then one of these xylulose 5-phosphates, the leftover one, reacts with erythrose 4-phosphate. And that completes the stage 3 of the carbon-carbon rearrangement steps. The net products that are obtained are fructose 6-phosphate, shown here, 2 molecules, and we get 1 molecule of glyceraldehyde 3-phosphate. Let's talk about the rate-limiting step of the hexamonophosphate pathway, and that's glucose 6-phosphate dehydrogenase. Now we're really entering into the stage 1, the redox phase of the reaction. This, as we mentioned before, is the rate-limiting step. It's allosterically activated by NAD+. It's very interesting is that there's a lot of etiology related to this enzyme, particularly in many populations and different ethnicities that seem to be targeted by mutations in glucose 6-phosphate dehydrogenase. Some mutations are innocuous. Amongst the more malignant mutations are those that cause hemolytic anemia, chronic hemolytic anemia. That is where the red blood cells begin to lyse. And one of the reasons why this glucose 6-phosphate dehydrogenase is important is because of that detoxification that you get by generating the NADPH reducing equivalent. If this enzyme is shot or mutated, or you have a particularly malignant mutation, you can't generate NADPH, which means that you can get a toxic buildup of free radicals and other types of intermediates, which can damage other parts of the cell. The enzyme is also linked on the X chromosome, so females actually have 2 chances of getting the right copy of the gene. Males, which are XY, have only 1 chance to get the right form of the gene on their X chromosome. People with glucose 6-phosphate dehydrogenase mutations or deficiencies are susceptible to the 3 A's. They are sensitive to antibiotics, they are sensitive to antimalarials, and they are sensitive to antipyretics, primarily due to the fact that the NADPH is not generated to allow for detoxification. One of the more weird forms of this mutated enzyme is this ailment known as favism. Favism is a sensitivity to the fava bean, which is particularly staple in a lot of Mediterranean diets. And people who have glucose 6-phosphate dehydrogenase deficiency or mutations, or bad mutations of the enzyme, for whatever reason, cannot eat fava beans because the ingestion of this beans generates free radicals, and the NADPH simply cannot handle that because the enzyme is not there that produces NADPH. Red blood cells do have a way to detoxify free radicals. that NADPH malate dehydrogenase serves as an alternative to get rid of free radicals because that form of malate dehydrogenase makes NADPH. And children who suffer from glucose 6-phosphate dehydrogenase deficiency are often subject to neonatal jaundice. People who have glucose 6-phosphate deficiencies, particularly that population that's in sub-Saharan Africa, they've actually, as with all malarial pathogens, seem to have a better resistance or better protection against the malarial pathogen Plasmodium falciparum. This is a selective advantage to have glucose 6-phosphate dehydrogenase deficiency because the red blood cell is in bad shape, and that means the Plasmodium falciparum can't get in there and complete its life cycle because the red blood cell is in bad shape because of this deficient enzyme. So somehow throughout evolution, a compromise was reached so that many people where malaria is endemic have their red blood cells that are glucose 6-phosphate deficient. However, these individuals are also resistant to Plasmodium falciparum and don't get malaria. So that's a very interesting evolutionary balance between two pathogens, the pathogenic status of this mutated enzyme and the actual parasitic pathogens. More normal populations that are susceptible to glucose 6-phosphate dehydrogenase deficiency that have not accommodated to this malarial parasite. One of the hallmarks is these Heinz bodies. These are occlusions of different types of materials, probably mutated enzyme and other types of free radical byproducts that have sort of accumulated in these different cells, and they're known as Heinz bodies. And that's one of the diagnostic indicators of deficiency of this enzyme. So the very first enzyme of the pentose phosphate pathway has some clinical consequences. These are all reversible, the carbon-carbon rearrangement reactions. The second phase is isomerization and epimerization, and then the initial phase is oxidation reduction. So here is the relationship between glycolysis and the pentose phosphate pathway. Notice here we're looking at the pentose phosphate pathway. If the cell wants to make DNA and RNA, it could bifurcate right here and make those nucleotides, nucleic acids. The bifurcation can also occur here and make xylulose 5-phosphate, which in turn can go and make through stage 3, the carbon-carbon rearrangement phase, fructose 6-phosphate and glyceraldehyde 3-phosphate. So it's important to realize that this can shunt into glycolysis to make pyruvate, or the fructose 6-phosphate and the glyceraldehyde 3-phosphate can be shunted upward to make glucose via gluconeogenesis. So the pentose phosphate pathway provides, it feeds into both glycolysis and the opposite of glycolysis, gluconeogenesis, and it can also feed into DNA and RNA synthesis. The bifurcation point occurs right